We're going to share with you some quite preliminary early results from a ESRC project, ESRC funded project that we have ongoing up here in Edinburgh, a project, as Emily suggested, titled The Life Course of Place. And it's a project that we've been working on with colleagues at Edinburgh in the Edinburgh College of Art, the Lothian Birth Cohorts, particularly Simon Cox and Ian Deary at the LBC, and colleagues in Creche, including Gergo and I, but also Neve Shorts, around a collective across Edinburgh University studied this. I'm just going to have a go at sharing a few slides. Bear with me. If someone could just shout at me, you can hear that because I can't see you anymore now.
Yeah, that's good.
Thank you very much. So yeah, Gergo and I are going to do a double act. So perhaps we'll just introduce ourselves quickly. I'm Jamie Pearce. I'm Professor in Health Geography up here at Edinburgh, and I'm also Editor-in-Chief of Health and Place and Wellbeing, Space and Society. So 2 journals to recommend to you all, especially if you're very interested in receiving work on life courses, longitudinal studies of health and place, for reasons we'll talk about in a moment. That in a nutshell is me. Gergo, do you want to quickly say hello?
Sure. Hi, everybody. My name is Gergo Baranyi, and I'm a postdoc working on this project with Jamie and with all other people. Just short to my background, I'm a psychologist by background and an epidemiologist, and I did my PhD in health geography, so mixed, basically. To you, Jamie.
Thanks, Gergo. So Gergo and I are going to have a go at doing a double act here. It's basically going to work. I'm going to do the easy bit. So I'm going to do the introduction and contextualize the study and tell you a bit about how it came about in the first place. And then Gergo is going to do the hard bit of telling you about some of the new analyses we've been doing over the last few months as part of the ESRC study. So we'll break into two bits and, as I say, split them up between us. So just to sort of contextualize our project a little bit, the project is intended and has been sort of situated within the broad healthy aging agenda, and I'm sure you're all familiar with the healthy aging agenda in its very broad sense and why it's become such an important global and national policy priority. For example, we're expecting to see a doubling of the population of people aged 60 and over between now and 2050, and a 3-fold increase in those aged 80 plus over the same period. So of course there's a big demographic shift, and also of course not everyone ages in the same way. There are very stark differences in physical and mental capacities, also in terms of functional abilities between people, between places. And a range of factors are likely to be critical to understanding healthy aging and why declines in health and well-being vary so much between people in older ages. And this piece of work is particularly on some of the place-based factors that potentially contribute to healthy aging, and particularly how place-based factors across the life course affect aging-related outcomes. And so we'll be talking in a minute a little bit about the sort of theoretical, conceptual rationale of this agenda, why hopefully it's offering something relatively new, but also it raises a whole suite of methodological data challenges, some of which we've hopefully managed to grapple with, but I think there are certainly many more for us to work through and develop even more appropriate, robust approaches in the future. So we'll offer our first go at some of these questions. So that's I guess the broad agenda around healthy aging. We could just take a brief step backwards for a minute. So as I said earlier, one of the things I do in my spare time is to edit Health and Place, and again, I'm sure this audience is particularly familiar with the sorts of things that Health and Place tends to publish, but if I was to try and pin it down into a very pithy summary, I guess there are two sets of papers or two sets of aspects of place which occur most frequently on the pages of Health and Place. Firstly, what we might think of in terms of the opportunity structures of place, the kind of opportunity structures in the places in which we live and we work and we play. So people looking at, for example, secure and non-hazardous employment opportunities, people looking at issues such as housing quality, people looking at green spaces, health and well-being, and various other outcomes. Other aspects of the physical environment, that might be things like air pollution, noise, or other sort of hazardous, risky environments, and also local public services, so the sorts of infrastructure that's available. So we might sort of think of these, as I say, as a kind of the opportunity structures. I'm sure you can come up with other examples as well. The other broad area is what perhaps the less tangible, the less sort of physical aspects of place, and what we might term the collective social functioning and practices of place. So this could include, for example, the local norms and values. What's a kind of normalized behavior? Is it normalized to go out running in the place that you live? Is it normalized to be a smoker, for example? We might also think about sort of social capital and networks in our local communities and how they translate into health outcomes. Neighbourhood reputation or stigmatization of place has been a sort of emerging theme over the last few years in terms of the work we've had coming in. And perhaps thinking some of the work that Gergo's done in the past, thinking around local crime and how that matters for mental health outcomes in particular. So I guess that's a sort of the type of thematic work we had coming into Health and Place. And as I say, probably no great surprises to people in this audience. I'm sure you've probably got your own conceptualization, but just as a sort of a starter there. But if we sort of look at all this work, it's certainly thematically diverse. It's theoretically, I think, collectively very rich. But I think there are some important critiques, which, again, I think will be particularly familiar to the land group. So I guess the first thing to say is the vast majority of the work that we have coming into the journal is cross-sectional. It relies on contemporaneous exposure assessments in some way. It's looking at often very contemporaneous health issues, and there's not a great deal of longitudinal work in the Health and Place area. I guess that's, as I say, sort of why this group I think is quite a timely and exciting group to be involved with. Or when there is longitudinal work, it's most ordinarily usually very short term. So it's maybe an evaluation of some sort of an intervention and or a natural experiment. There might be a pre and post analysis. If we're lucky, we might get 2, 3 or 5 years follow up to look at the effect of changing some aspects of the environment, and then how that has or hasn't translated into affecting health outcomes. Usually some quite short-term evaluations. But what is, I think, generally missing from the pages of Health and Place, generally, with some very notable exceptions, people gathered, mostly people from in this meeting, there's not a lot of work examining environmental circumstances over the full life course, and alongside this, an oversight in terms of thinking about how historical context, how particular era-specific aspects of place are important for understanding contemporary health outcomes. So I guess that's sort of a broad critique, which, as I say, probably isn't a huge surprise to this particular group. And it's with that backdrop in mind that our current ESRC-funded project came about, a project entitled Life Course of Place, How Environment Throughout Life Can Support Healthy Aging. And I've already mentioned some of the collaborators working with Gergo and I on this ESRC-funded study. The questions or the challenges that we posed to ourselves in this project was to think about, firstly, is it feasible for us to develop a longitudinal approach to examine the connections between health and place across people's lives? Is this a feasible particularly data, but also conceptual tasks. Can we integrate historical environmental data with cohort of individuals covering full life histories? And also, is it possible for us to reconstruct, inverted commas, past urban environments? So is it possible to identify and bring together the data that we need to answer these types of questions? And if it is possible, can we therefore answer questions about whether there are accumulative effects of place? So is it that the kind of environments that we face and experience through our lifetimes in some way accumulate over time, and then there's a net result at the end of that in terms of our later life health experiences? And or are there critical periods in the life course where places are particularly pertinent? So is it possible that, for example, in childhood, that those sorts of experiences, those sorts of environments are so critical because they represent a formative moment in the life course, that those experiences in childhood affect our health regardless of what happens going forward through the rest of our lives? So that's what this diagram's trying to visualize, whether there's a cumulative effect or these particular critical periods. So those are the sorts of questions that we were trying to get at. As part of this, we've been talking about this life course of place approach, which builds on lots of work in particularly in human geography, but I think across the social sciences, health sciences as well, around the connections between health and place, and thinking about why this conceptualization potentially matters. I guess the first thing to say is, again, very a kind of a message that will be very familiar to the geographers in the meeting today, that places, of course, are socially produced, they're dynamic, they change over time in response to all sorts of structural, political processes, the different sort of power relations that we know matter in terms of shaping place. As I say, reflect sort of the macro level processes that often operate over many decades. And of course, we can see this up here in Scotland and all around the country, as there's been very significant changes in terms of not least the sort of industrial landscape and the way in which that has played out in our cities and affected the urban infrastructure, the opportunities that people have, and also the kind of environments in which people live. And so I guess what that tells us, of course, is that environmental and social differences are inextricably connected. At the same time, of course, as geographers, we know that local particularities matter. They mediate relationships through local resources, rules and practices, and can reinforce and rework place-based characteristics. So it's kind of just in a nutshell, just trying to bring together some of the kind of the writings in human geography by many people over a number of years, and sort of thinking about this in a specifically health and health equity kind of lens. So how do we go about this? How are we doing this? So thinking about some work we've been doing for a little while, and this is where the new project, the current project, emerged from. We've been working with colleagues here at Edinburgh who run something called the Lothian Birth Cohort. And the Lothian Birth Cohort grew out of a survey that was sat many years ago by children aged 11 in 1947, so children born in 1936, and then age 11 they set this the mental survey test, which is intended as a test of intelligence for 10 and 11-year-olds. And this data was collected through a series of what are surprisingly difficult questions if you run through the survey aimed at 10, 11-year-olds, but certainly I found it quite challenging to try and go through. But maybe that's just me. But these tests were sat, and then largely sat in a dusty cupboard in part of Edinburgh University in the education school, Moray House of Education, for many, many decades before being rediscovered back in the early 2000s. And some of our colleagues in the Lothian Birth Cohort, particularly in Deary, instrumental in revitalising them and recontacting some of the participants, particularly the participants living in the Lothian region of Scotland, which is kind of the Edinburgh region of Scotland. So the test was sat by around about 70,000 kids aged 11 across Scotland, and those living in the Lothians were followed up. And that's what this slide is giving a bit more detail on here. Participants followed up at age 70, age 73, 76, 79, 82, and currently, or very recently, have been in the field over the last year or so as well to add a sixth wave, although that data is not quite ready for analysis yet. You see there's just over 1,091 participants were originally agreed to participate in the study. So we had some really rich detail about them aged 11, and then a whole suite of information. They were prodded, tested, and examined and filled in multiple questionnaires about all aspects, many aspects of their health and their well-being, but also demographic, family history data, and much more besides through different waves of the Lothian Birth Cohort. And there's a cohort description paper if people are interested in knowing a bit more about the Lothian Birth Cohort. But what the Lothian Birth Cohort didn't have, which was important for our perspective, was anything about where people lived and where they worked through their lives. So for a study that was trying to look at context, about geography, about the role of place across the life course, of course it was important to have that information. So what we did was to use something called the life grid technique, which has been reasonably well utilized and pretty well tested out previously. What it does is it uses, as you can see from this this partially completed form that all participants were provided with, it provides a series of memory prompts about particular events in history. So this one from the 70s is talking about the oil crisis, when Thatcher became Prime Minister, in the 80s, the Falklands War, the Lockerbie bombing, which of course was a very major event here in Scotland, Hillsborough disaster, and so on. And it helps people to then recollect where they were living at different decades in their lives. And so this was certainly very successful from a data completion perspective, in the sense that the participants reported they were confident and were able to fill in the form. But of course, as we'll talk about later, that does potentially raise some recall bias issues that we certainly at the very least need to be aware of. But nonetheless, gave us some rich information about where people were at different points in time. And we were able to go back and then, of course, geocode that information, which wasn't, I should say, a trivial task, given that especially in urban areas, some of the larger urban areas of Scotland, many of the areas where people identified as living no longer exist because they were cleared often as part of the post-war slum clearance programs, the redevelopment programs of the 1950s and 1960s. So we can say more about that if you're interested, but just to say it wasn't a trivial task going back and geocoding the data. So that gave us some information about where people lived, about the individuals, and we obviously had rich information about them from the waves of the Lothian Birth Cohort and the original survey they sat in the 1940s. But of course, what was still missing was information about the places that people lived through their lives. And so what we did was, this was in our original work a few years ago, we spent a lot of time in the various archives around Scotland, particularly in Edinburgh, digging out various reports and other paper-based, largely paper-based material, such as the civic plan, the City of Edinburgh, which was the post-World War post-World War II reconstruction plans, which contained a really rich source of geographically specific information that we mined and pulled out all sorts of information related to housing and all sorts of aspects of place. Another example of a resource we used was the Sanitary Department annual reports for Edinburgh, which each urban area in Scotland had to provide annually right through until the 1960s, 1970s. So again, gave us some really useful social, demographic, and of course health-related data for neighbourhoods or areas across the city. So we mined this information and pulled out all sorts of data. Here's another example. This is some information we pulled out around public parks in Edinburgh at different points in time through the 20th century. And it shows, I guess, rather obviously the growth, the increase in number of green spaces, pretty much reflecting the growth and expansion of the city. But also, if you can sort of pick out, for example, some of the red dots, areas that were public parks at one point in time had disappeared by the subsequent period of time, so where often the public parks were built for, or land use was changed towards housing. So yeah, you can still see how we can start to build up, give us a sense of the changes in places over time. If you're interested a bit more about the methodology for this and the broad sort of application for life course epidemiology, then there's a paper we published quite a while ago now, if you need help getting to sleep. But what that allows us to do is, of course, to build up a picture at the individual level. So here is a sample of Lothian Birth Cohort participants, originally numbered from 1 through to 1,000 and whatever. Here's a sample of them. And you can sort of see from this heat map the kind of green space experiences that they had through their lives, or have had through their lives. You can see, for example, say, you can zoom in there and you can see that LBC 131, participant 131, you can see that at the start of their lives, they lived in a relatively green part of the city, but then moved into or the place around them changed possibly into a less green place, and then they're sort of somewhere in the middle for the rest of their lives. Other participants, such as some of those towards the bottom, you can see early in their lives, they lived in areas of relatively low green space, but by their 70s and 80s, they were living in some pretty green parts of the city. So you can, this is just one variable. Of course, you can apply this to a number of the other data sets that we've brought together. So the questions just to give you a very quick sense of some of the initial analysis that we did as part of our previous work. We did some work looking at green spaces through people's lives and the effects and associations with cognitive aging related outcomes, and also green space with mental health outcomes, asking these questions about accumulative or critical period effects. So I'll just say, run through this really quickly, but one of the pieces of work looking at green space and cognitive aging was, I think, quite interesting. And what it showed was that, perhaps unsurprisingly, there was no association between green space and the change in cognitive test score between when they sat the test as a child, aged 11, and when they first entered the Lothian Birth Cohort, aged 70. And that's no great surprise, because there isn't a lot of change in terms of cognitive ability over that period. But where it did get a bit more interesting was when we look at that cognitive change after the age of 70, where some people do start to see a decline in cognitive test scores. And it did show, in that case, a link with green space, and in particular showed that childhood seemed to be a particularly sensitive period, affecting cognitive function trajectory in later lives. In other words, the types of green environments that our participants lived in early in their life in childhood seem to have lifelong implications in terms of their cognitive changes in their cognitive score after the age of 70. And we also looked at this in terms of mental health and some quite dissimilar results, really. We looked at an outcome, one of the outcomes available through the LBC, Hospital Anxiety and Depression Scale, and a subset of it, the anxiety outcome. And we saw some similar things, although the influence seemed to be much more limited to the most socially disadvantaged groups. But again, it seemed to be suggesting that green spaces in early life tended to impact upon mental health in later life, even after you account for the other things that you think could potentially confound that relationship. So that was our sort of, I guess, our pilot work. And what it led us to do is say, well, that's been really useful, but we're not really taking advantage of some of the major strengths in the Lothian Birth Cohort, or indeed actually looking thus far at a broad range of environmental characteristics. So that's where this new SRC study came in, and what we looked at or are looking at is whether lifetime exposure to green space, air pollution, and area level deprivation, we've largely focused on that so far, affect quite a broad suite of indicators of healthy aging. So we've got that is, I say, playing very much to the strengths of the Lothian Birth Cohort, which does have very rich information on cognitive aging and healthy aging more generally. So our measures, as Gergo will say a bit more about in a moment, we're looking at the various indicators of cognitive aging. We've got information on brain health collected through various scans and other approaches, and also measures of sort of biomarkers of biological aging, which we might talk a little bit about later. And once again, looking at these the impact of potential critical periods and all these cumulative effects on brain health and other cognitive outcomes across the life course. I think, Gergo, this is where I pass over to you. So give me a shout when you want to move on slides.
Okay, thank you. So as Jamie mentioned, in this new project, we are trying to focus on how basically neighborhood can get under the skin. So we are not focusing anymore on major health outcomes, but more like different aging phenotypes or like markers of biological aging. So in the first study, which I would like to present you shortly, is about neighborhood social deprivation and frailty. Next slide, please. And we know that frailty is, thank you. So we know that frailty is, well, it's basically an age-related syndrome, an age-related like phenotype, which is associated with increased vulnerability, loss to resistance to stressors, and with decreased reserves of capacity. Basically, more frail adults, they have lost ability to recover from injuries such as the fall, and they have also lost ability to recover from illnesses and diseases. And we also see that frailty generally develops across a longer period of time and across different domains of functioning, such as psychological functioning, cognitive functioning, and so on. Next, please. We have also seen that frailty has a very complex etiology, and recent studies suggested that also neighborhoods with physical and social features can be linked to frailty. However, as you might also assume, there is very limited knowledge on the longitudinal association between frailty and neighborhood, and between frailty and neighborhood features. And we couldn't find any previous study which would have applied the life course approach. Next slide, please. And this is exactly where our study tries to bring something new. So we try to apply the life course framework by establishing the association between neighborhood deprivation and frailty, and also frailty progression, because we have several ways of frailty measurement. The next slide, please. So just to put everything into a timeline and see how all this data comes together, here you can see that participants in the Lothian Birth Cohort 36, they were born in 1936, age 11, they set the Scottish Mental Survey, and then from 2004 onwards, they have been followed up 5 times. At the very first wave, approximately 1,100 individuals at average age of 70 were participating, and in the last available wave, approximately 430 individuals at the age of 82. Next one. So can you go to the next slide, please? Perfect, thank you. And then, so neighborhood deprivation was assessed across the life course for each decade using historical data, as Jamie already mentioned. And after that, we used fractal analysis to identify particular exposure periods. And we saw that actually there are 3 exposure periods which can be like distinguished. One, neighborhood deprivation in childhood, another one, neighborhood deprivation in young adulthood, and then neighborhood deprivation in mid to late adulthood. So this was exposure. In terms of the outcome, we used the so-called frailty index, which is a continuous measure representing accumulation of health deficits. And it has been developed from 30 different variables across physical, psychological, and cognitive domains. So the values of frailty score ranges between 0 and 1, And certainly, as you would assume, over the time, the mean value becomes larger. The next slide, please. And we used covariates also across the life course, such as, for example, childhood covariates as age 11 IQ or childhood smoking, occupational social class, and also covariates from late adulthood. Next slide, please. And in terms of deciding which life course model might be the most appropriate, we applied the so-called two-stage structured life course modeling approach, which has been originally established by Mishra and colleagues in 2009, and then later modified for continuous exposures by Smith and colleagues. So just very shortly, this approach in the first stage tries to identify the best fit life course model between several competing ones, such as sensitive periods or accumulation and so on. And here it applies the least angle regression algorithm, again, to select the life course model, which is the strongest associated with the outcome. And in stage 2, so next slide. So in stage 2, then we use the selected model to estimate its impact on the outcome in a regression framework. For this one particular study, here we estimated the impact of selected life course models for frailty at age 70 with linear regression, and also for the frailty progression between age 70 and 82. The next slide. And the next slide here shows the results for the stage 1, so for the model selection approach. We run all models separately for males and females. So for the male subsample, we can see that actually this accumulation, which is the most appropriate life course model in terms of frailty, and we found that approximately 7% of the residual variance was explained by accumulation, by accumulating neighborhood deprivation. In the female subsample, we found that mid to late adulthood sensitive period was as the first selective life course model. However, it wasn't significantly better than a model without it. So next slide. So after selecting these life course models, so after selecting accumulation and mid to late adulthood sensitive period, we also estimated their impact. And you can see this on the graph on the right side. So in the male subsample, we basically found that those having a higher score in accumulated neighborhood deprivation, they were more frail from the beginning of the study. And then, although there was a little bit of narrowing across the follow-up period, we saw that basically they had also at the end of the study a higher risk of becoming frail. When we are talking about accumulation, certainly it's also interesting to see what part of the life course is more stronger associated with accumulation. And in a post hoc analysis, we find that especially childhood and mid to late adulthood deprivation was important in the male subsample in terms of accumulation. Well, in the female subsample, we saw something completely different. So while at the beginning of the study, so at age 70, we didn't see any difference in terms of neighborhood deprivation and frailty. So in individuals in more deprived areas, they had the same level of frailty as those in less deprived neighborhoods. However, as the time progressed, we saw actually growing inequalities in terms of frailty progression among those living in deprived and less deprived areas. So basically putting all this together, you can also see here that neighborhood deprivation is actually important across the life course. So it's not just the exposure where at the moment we are living, but also before, such as in childhood. And such childhood seems to be a particular sensitive, a particularly important period in terms of exposure. The next one, please. And in the next, exactly. So we have we already published the preprint for this paper, which is available online, and the paper is under review at the moment. So the next study, next slide, is try to dive into more detail and try to understand how exactly neighborhood exposures can go under the skin. And here we are focusing on air pollution and epigenetic aging. Next slide, please. So if talking about air pollution, I think we are not really surprised that air pollution is associated with a lot of different health outcomes. It is one of the biggest threats to human health. And also it is associated with significant economic burden. So previous studies have already established that long-term air pollution can be associated with cardiovascular diseases, with cancer, but also newer research suggests that neurodegenerative and mental disorders are associated with higher air pollution exposure. The next slide. And one of the ways how air pollution may affect health and well-being is epigenetic regulation. So this is one of the underlying biological pathways, certainly in addition to oxidative stress, inflammation, and so on. When we are talking about epigenetic regulation, we understand under it mechanisms. They regulate actually gene expression and gene transcription without changing the gene sequence itself. So one of the epigenetic regulatory mechanisms is DNA methylation. And DNA methylation basically describes a process where when a methyl group, which is a CH3, when a methyl group is added to the 5th position of a cytosine molecule in the genome. And by adding this methyl group, actually the gene expression changes. So it can actually offset a gene or like change the activity of a gene. And certainly these cytosine molecules, they are usually sitting at so-called CPG sites, so like cytosine phosphate guanine sites. The next one. So research has already seen or like already established that individuals exposed to adverse experiences like higher stress or accumulative stress across the life course, for them we have seen that actually there is alteration in DNA methylation, in particular CPG sites, there is hypermethylation or hypomethylation. And also actually methylation patterns can be observed in the occurrence of several diseases, such as, for example, cancer. So individuals with cancer, they have very often a higher rate of methylation across the genome. Also, actually, during aging, specific regions of the genome become methylated, which actually also actually can help us to identify specific regions in the genome, which might be more linked to aging. The next one. And this also led to very recent developing so-called measures of biological aging or measures of epigenetic aging. And next one. So here, particular like epigenetic clocks have been developed in the last years using, for example, Horvath's epigenetic clock or like Hannum's epigenetic clock. These are like 1st generation epigenetic measures. And the next. And then also pheno-age or Grimm age or DNA methylation age. These are 2nd generation epigenetic clocks, which have been recently developed. So next one. One more. Yes, thank you. And then here comes again, or like research objective for this particular paper. We again try to understand how air pollution across the life course, the exposure to air pollution across the life course can impact epigenetic aging. Next one. Because we already see in several papers that there are some links between epigenetic aging and air pollution. However, these are very often focusing on shorter follow-up time. Yes, just some very short information about how the data comes together. Again, for DNA biomarkers, we only use data from the first 4 waves, and we conducted. So we calculated 5 biomarkers. Again, the Horvath, Hannum clocks, and pheno-age, Grimm age, and DNA telomere length. Next one. In terms of air pollution, we used historical measures of air pollution exposure using the models developed by colleagues here in Edinburgh. These air pollution concentrations were calculated for 1935, 1950, 1970, 80, 1990, and 2001. So basically covering the entire life course of the LBC sample. So concentrations were available on a spatial resolution of 5 to 6 kilometer grids. And we had values or concentration values for PM2.5, SO2, NO2, and for ozone. Because these pollutants were very highly correlated, and when I'm talking about high correlation, they were very often correlated on a 0.9 level. And that's why we decided that we will calculate a so-called cumulative air pollution index. So in order not to overtest our hypothesis. So we used in this paper cumulative air pollution index instead of each of the pollutants. Next one. In terms of the analytical approach, we used the exact same approach as in the previous paper, so I wouldn't go into more details in that, but show you immediately the results. So what we saw in the model selection stage, we tried to identify the best life course models out of, as you see here, 7 life course models, so sensitive periods in each of the exposure types and accumulation. So basically we saw that that exposure in 1970, air pollution exposure in 1970 was associated with Horvath's epigenetic clock in the total sample. In sex stratified samples, we found that exposure in 1980 in the male subsample was associated with DNA methylation-based telomere length, and in the female subsample, in utero exposure was associated with DNA methylation-based telomere length. Go to the next one. And after selecting these hypotheses, or selecting these life course models, we also estimated their association with the different epigenetic clocks, different epigenetic measures, which you can see here in the figure. So just a few words about the figure. We are showing here standardized coefficients. So we standardize both exposure and outcome in order to compare them a little bit better. And in addition to the cumulative air pollution index, we present here also each of the air pollutants, so PM2.5, SO2, NO2, and ozone. So in the very first part, you see the results for Horvath's epigenetic clock. So basically, you see that actually higher exposure to air pollution in 1970 was associated with accelerated aging using Horvath's epigenetic clock. However, as you can also see for O3, so for ozone, the association was not significant, but it was in the other direction. It is basically because ozone is always negatively associated with other air pollutants. In the middle figure, you see that DNA methylation, so it's actually presents DNA methylation-based telomere length. Again, here we see actually that shorter telomere lengths were observed among those with higher exposure to air pollution, and certainly shorter DNA methylation telomere lengths indicate actually accelerated aging, so faster aging. And similarly, also in the female subsample, we saw that higher exposure to air pollution in 1935 was associated with shorter telomere lengths, again indicating faster aging. In all of our models, we also controlled for false discovery rate adjustment, because we test a lot of models. In this case, you have to control for type 1 error. And we saw that actually the most robust results were based on air pollution in 1936. So the very last bit of this figure is the one which is potentially the most robust, indicating actually that air pollution in utero has very long consequences on biological aging, which can be even observed 80 years later. Okay, I think that was about my part, so I'm back to you, Jamie.
Yeah, thanks, Gergo. And just because we're short time, what I think I'll do is I'll just jump ahead a little bit, so we've got some time for discussion. This was just really to sort of think about the conceptual contribution around this, but I think that's probably well known amongst this group. Just briefly in terms of limitations, I think we've already talked about recall bias, particularly in relation to the life grid technique. Also, we need to think about survivor bias, or any cohort of healthy aging, but including the LBC. Also, this is quite a low mobility population. We focused on people here who have been in the Lothian region of Scotland. So these are, of course, by definition, are relatively low mobility people. And also there's certainly issues with each of the kind of area level measures we've talked about in terms of the ideal data that we would have wished for, how the meaning of different variables change over time, and also issues around reporting units. So there's definitely work to be done there. And I guess conceptually, I think we also need to recognise that we probably could usefully draw on greater methodological innovation from the social sciences that capture something more than the static snapshot of places, when of course we know that places churn and evolve, and capturing some of this I think would be another useful conceptual step forward from what we're offering here. Just briefly in terms of next steps, we're looking at a wider set of environmental characteristics. We've seen pollution, but broadening that still. The new set of outcomes, cognitive aging, brain imaging, and then you've already heard about the biological aging. And then we're also involved in some other work that's trying to scale this up nationally, at least here in Scotland, using this close longitudinal study, potentially a British cohort. Naomi and I have talked about this for a number of years, which would, I think, need some greater methodological innovation in terms of how we capture some of the area level characteristics, perhaps auto extraction mapping techniques, which we've been piloting as one possibility. And I think also international comparative work would be really great to test out some of these ideas in other contexts.